Hello students, welcome to MOOC course on Introduction to Proteogenomics. We are now in week 2. So welcome to the second week of this course. So as you're already aware, this course is 12 weeks long and you have finished successfully the week 1 and assignment 1 activity. I hope you got a chance to interact with us on Saturday on the Skype call. Many students have taken the slot to discuss their queries and their questions face to face live with our team. So please utilize this opportunity for interaction with us. And if you are finding anything difficult to follow or some concepts are unclear, I think good idea to start interacting with us from the beginning. Don't wait for a lot of things to get accumulated and then only ask questions. So this week, Dr. Kelly Ruggles, she will continue her lectures and she will talk about gene regulation and epigenetics. If some of you are core into biology, you know the significance of epigenetics and chromatin remodeling and how some of these are very important for different diseases, especially cancer. Uh, she'll also talk to you about chip sequencing and introduce you to some of the publicly available database. Uh, for example, the TCGA in code. Some of these databases can be utilized to obtain large data sets from different type of cancer projects. And also you can start utilizing and analyzing that for your practice as well as for your actual projects in future. Kelly will then introduce you to the another important portal, which is the CBio portal for genomics analysis. Again, a lot of cancer data sets are available and how to access those. That will be introduced to all of you. So I hope with some of these lecture by Dr. Kelly Ruggles, you are now getting comfortable to know that there are large resources available publicly, which you can utilize for learning as well as for doing research, especially in the area of cancer genomics. So Kelly Ruggles, along with her, now we will be having another, instructor in the course, Dr. Bing Jiang from Baylor College of Medicine. So both Kelly and Bing Jiang has been actively involved in the CPTAC program, which is a large consortium for doing proteogenomics cancer research, and they are very, uh, you know, uh, enthusiastic scientists who are coming up with very novel ideas of integrating a huge amount of data sets from genomics and proteomics and build the pipeline for doing proteogenomics work. So you're very fortunate that you are really learning some of the best tools from the people who have developed these actually, rather than just you are trying to understand from somebody else. So these are the key contributors who are part of these projects. And Bing Jiang will talk to you about some start from very basics about looking for genotype, genome analysis, talking about variants, and then he will talk about QTL analysis from cis and trans. He will then continue about some more computational tools to analyze QTL studies and integrate that with GWAS studies. All of those are very important for the functional genomics and to really gain insight about a biological system. So some of these tools will be very helpful for you to get introduced at early stage now, because as you can understand, these are very key tools for understanding the biology and especially for understanding different diseases. If you are aware of these different resources, you can really utilize them well for different projects. Finally, for this week, the lecture 10th will be by Dr. Atima Agrawal from Thermo Fisher Scientific, and she will talk to you about next generation sequencing technology. Next generation sequencing will have more instructors and more guest speakers who will talk to you. She will introduce you about ion torrent technology and start with the basic concepts of this, as well as some of the steps involved in doing NGS like library preparations and all. So some of these things, look slightly advanced at some stage if you think about, but I'm sure you will really enjoy learning the most advanced tools, most cutting edge tool, sitting at home, learning from the best instructors of the world, and also clarifying the doubts from IT Bombay team. So enjoy your week 2 activities, and we look forward for your assignment 2 for the coming week. And also, In this video description, you will find a lot of details for the reference material, for your form which you can fill to take a slot to talk to us on Skype on Saturday. And of course, all the video links are there. So all the best for the week 2.